Hello everyone! I would like to welcome you to our third series of your microbiology class with our title, Microbial Diversity, an in-depth study of your acellular cells. For our past two discussion, we are repeatedly saying the word acellular. That's why I will presume that as of the moment, you have a sound understanding of what acellular cell is. However, I would like to describe to you what acellular microbes are. Technically, these microbes lacks the basic characteristic of life, which is your cell. They usually are microscopic by nature, and as a result, it is significant for us to somewhat study these microbes because they bring about significant changes to your various host cells. When we talk about acellular cell, the word virus should always come into mind. In order for us to understand what viruses really are, we need to define what virions are, or virions, however you pronounce it. Your virions are basically the term we use to denote a complete virus. Meaning, lahat ng characteristic, all the physical traits and characteristics of your virus are present to these virions. In order for us to visualize viruses, because of their so small dimension, you need a special microscope. I'm talking about your electron microscope in order for us to visualize these microorganisms effectively. Also, it's a common knowledge that there is no particular organism that is safe from any viral infection. Well, at this chapter of our discussion, I would like you to accomplish a mini assignment. This will serve as your attendance as well as a form of formative evaluation if you have indeed learned something from our discussion. The instructions are posted in this PowerPoint presentation. Let me just read. You are to list 5 diseases that is considered as viral infection. Also, you are to indicate the name of the disease together with their causative virus or the causative agent. Let me give you an example. For example, this current pandemic, COVID-19, that is the disease, and the causative agent will be SARS-CoV-2 or your SARS coronavirus 2. Ganun lang yung gagawin ninyo. I need 5 examples of this. You cannot use the example that was already given. Further instructions will be provided to you using your canvas. In order for you to understand what viruses really are, you need to find or understand these 5 properties of viruses. Number 1 is that it should have a genetic or a hereditary component. I'm talking about either a DNA or an RNA. Hindi katulad ng mga eukaryotic cell or other cells, for that matter, your eukaryotic or prokaryotic cell or your cellular microorganisms. Acellular microorganisms might either have an RNA or a DNA. Hindi pwedeng both. Except for mimivirus, which will be discussed later on as we proceed with our discussion. Next, they are unable to replicate on their own. In order for them to replicate, it is needed for them to invade a particular host cell. That's why they are said to be obligatory pathogens. Kasi hindi sila mabubuhay nang walang kinakapitang iba. Next will be, in order for them to reproduce, they do not use or they do not undergo binary fission, mitosis, or meiosis. Later on, how they reproduce or how they increase in number will be discussed in this particular session or in this particular discussion. Next, they basically lack genes and enzymes in order for them to produce energy. That's why they usually depend on metabolites, enzymes, as well as ribosomes of their host cell. Okay? That technically makes them unable to sustain life on their own. Ergo, the term acellular. To study the viral structure, Again, as stated a while ago, every single virus will have their genetic material, either an RNA or a DNA. If you still remember our discussion, I'm not just sure if it's on our first or second video or even both. We have defined or illustrated an acellular cell as if it's a gift. Inside the gift is your genetic material, again, either your RNA or DNA, wrapped in a protein wrapper. This protein wrapper is called your capsid. Both of this nucleic acid plus your capsid is termed as your nucleocapsid. In order to produce new virions, they basically invade host cell in the because they do not have any ribosomes. Again, the very main reason why they are considered obligatory pathogens. In order for us to further classify your viruses, we need to look for the following characteristic. Number one will be the type of your genetic material. Next is the size and the shape of your capsid, the number of capsomeres. I forgot to mention, capsomeres are found on your capsid, doon sa outer covering. Next will be the presence of an envelope, your type of host cell that they basically infect, the diseases they produce, their target cell, as well as their immunologic or antigenic properties. We start with your genetic material. Pag genetic material, unang papasok ang two words, RNA or DNA. There are basically 4 classifications of viruses according to their genetic material. They can either have a double-stranded RNA, a double-stranded DNA, a single-stranded RNA, or a single-stranded DNA. Most of the times, the genome or this DNA and RNA present in your viruses are arranged in a circular pattern. However, at times they can also exist in a linear pattern. Classifying our viruses further will be basically from their size and shape of their capsid, or what you call the morphology or the viral morphology. Looking at our PowerPoint presentation, there are basically 5 classifications of viruses according to the shape or the size of their capsid. Number 1, you have your polyhedral. Pag sinabing poly, marami, or in English, numerous. Ibig sabihin, they have multiple sides. More than 20 is what you call your or icosahedrons. Next will be, it can either be seen on a helical tubular shape or coiled tubes. They can also exist in bullet shape form, spherical shape, or even combination of all these shapes. Each facet or side of this particular capsid will contain your capsomeres. The number of your capsomeres and the size of your capsid will basically determine the total size of your viruses. Also, you need to understand the concept of a viral envelope. When we say envelope, it will be basically comprising the outer covering of your virions. Usually, this envelope makes our viruses appear spherical in shape under a microscope. These are basically acquired during the process of budding. Mamaya, explain ko kung ano ang budding. However, I would like you to understand that your envelope are basically components, either component of your cytoplasm or your nuclear membrane of the host cell, where your virus or your virions propagated. Okay? Viruses are able to alter this particular membrane, this envelope, covering them, having proteins on the surface that serves as your receptors or your knobs in order to detect different receptors. This is the very main reason why, if you will observe coronavirus from the term itself, corona, it resembles a crown. Under a microscope, you will be able to see that this particular viruses will have like these crown-like structures or crown-like spikes present on their envelope. These are basically protein substances that got stuck on their envelope. Okay, this will now serve as knobs or something that will signal presence of a particular receptor from a host cell. Parang signaling capabilities. They will determine if the virus is compatible with this particular host cell. Now we talk about the origin of our viruses. Saan nga ba nagmula ang ating mga virus? We will talk about basically three theories that might give us an answer. Number one, the theory of coevolution. According to this theory, there is a particular bowl of primitive microorganisms, namely your bacteria and your archaea, and together with them are your viruses. As time goes by, all these three microbes co-evolved with each other. That's why it's termed to be coevolution. Another theory that explains the existence of these viruses is the theory of retrograde evolution. Pag pinag-usapan nating retrograde, from a particular cell, it evolved backwards as if it rewind. Okay? According to my lecture, this retrograde evolution explains that viruses evolved from free-living prokaryotes, your archaea and bacteria that invaded a particular living organism which later on lost its function. Therefore, the existence of your viruses. Next is your escape gene theory. According to this, when bacteria infects your particular host cell, they will leave several fragments of your DNA as well as your RNA. And this DNA and RNA will not have cellular control or will not be under cellular control, and later on they develop to be your viruses. Note, virus basically lacks the primary structure of life, which is your cell. That's why on their own, they cannot sustain life. Kaya nga again, tinawag silang acellular cells. Next, we're gonna talk about your bacteriophage or your bacteriophages. Your bacteriophage, let's somewhat separate this word. First word is your bacterio, pertaining to your bacteria, and phage pertaining to your viruses. That's why it's literally translated as viruses infecting bacteria. Okay? These bacteriophages are basically classified according to the processes they undergo after they have injected their hereditary material. There are basically two major classification of your bacteriophage. You have your virulent as well as your temperate phages. Mamaya, di-discuss muna natin sila. However, I would like you to understand that all these viruses that we are about to discuss requires a host cell in order for them to thrive. Again, the very main reason, that's why they are termed to be obligatory pathogens, because without this particular host cell, without them going to this particular host cell, they will die, they will cease to exist. That's why they need these host cells. Also, there are particular shapes that this bacteriophage can present. Looking at our PowerPoint presentation, we have this what you call complex structure. But before you understand the complex structure, tignan mo muna yung head part. That's what you call icosahedral or icosahedron structure, which basically contains multiple faces or multiple sides of a capsid. Also, you can find in the tail your filamentous shape, where it's described to be a long tubular structure of capsid proteins assembled in a helical structure. Together, when these are combined, this is what you call a complex structure, which is the typical characteristic or typical presentation in terms of shape of our bacteriophage. The first among the major classification of your bacteriophage is your virulent bacteriophage. When we say virulent bacteriophage, it will basically pertain to bacteriophages that will initiate the lytic cycle. Sir, ano po ba ang lytic cycle? From the word itself, lytic, ibig sabihin lysis. because the end product or the end result of this cycle is the destruction or the lysis of your host cell. There are several processes that you need to undergo in order for you to complete your lytic cycle. Starting with the first step, your attachment phase, or also known as your adsorption phase, wherein this particular viruses will attach themselves at the cell wall of your host cell. Take note, there is this term you call specificity, meaning phages or viruses can only attach to bacterial cell that contains a certain cellular receptors or appropriate receptors. Ibig sabihin, hindi basta-bastang nag-a-attach kung saan-saan ang mga bacteriophage natin. Kailangan that there is a specific receptor. Parang lock and key model lang yan. Hawak ng ating bacteriophage ang key, naghahanap siya ng specific na lock or your receptors. After attaching themselves into our cellular wall of our host cell, the second step will be penetration, wherein the bacteriophage will now inject. Pag sinabi nating inject, parang karayom, pag ini-injectan ka, hindi naman buong syringe pumapasok sa iyong deltoid. Rather, you are only injecting the content of the syringe. Same is true with bacteriophages. You are only inserting the content or the hereditary material present in this bacteriophage. This is the process wherein you will start to introduce and inject your DNA or RNA to a particular host cell. Now, this injected DNA will take over your host cell. It will take over with the machinery, the processes happening intracellularly. Next will be the process of biosynthesis. Now that the DNA has the control on your host cell, it will now tell the host cell, hey host cell, let's start producing the parts of our new virions. That's why we proceed to the third step, which is your biosynthesis, described to be the phase wherein phages will now use the host enzymes, amino acids, proteins, and ribosomes in order to produce new viral DNA as well as ribosome in order for them again to thrive. This will now serve as your recipe in order for you to proceed in the fourth step, which is basically your assembly, wherein these recipes, these ingredients will now be assembled in order to form our final product or our new virions. This process is where this viral DNA will be packed together in order to form virus with capsids. Kompleto na. After you have finally manufactured complete virions, it will cause the explosion or the destruction of your host cell. Ergo, our fifth step for your lytic cycle, the phase of release, wherein the host cell bursts open and new virions will be set free in the environment. That basically is your lytic cycle. Next type of your bacteriophages are your temperate bacteriophage. Pag sinabi nating temperate, ibig sabihin mahabang PC, mahabang pasensya. Basically, they were termed to be temperate because they do not initiate the cell death of their host cell or lysis. Hindi sila pumupunta sa lytic cycle. Rather, they now proceed to your what you call Lysogenic cycle, wherein the temperate phages will be injected to your bacteria, and now the DNA of this bacteriophage will disintegrate or integrate together with your bacterial DNA through the process you call transduction. Nagsasama, nagko-combine yung galing kay bacteria at galing kay virus. Now, this is the process you call lysogeny, a situation in which a phage's genome is present within the cell, the host cell, without resulting to its lysis or death. This host cell will start to divide, will start to produce their offspring through binary fission. While they are producing their offspring together with the bacterial DNA, viral DNA are also being reproduced. Now, there will come a time that this host cell will now be faced, or a particular extreme stimuli will be destroying or causing the host cell's lysis. When this will happen, those bacteria complex na DNA will now spread all throughout the adjacent cells, infecting the adjacent cells by either the DNA of that particular bacteria or the DNA of the infecting bacteriophage. Pipili lang sila. Ganon nangyayari ang ating lysogenic cycle with our temperate bacteriophage. The next type of your virus is your animal virus. Based from the word itself, these are viruses that basically infects animals and humans alike. There are several factors or several steps in order for them to successfully infiltrate their host cell. Almost similar sa bacteriophage pero may significant changes. That's why we need to discuss them as of the moment. Everything starts with step number 1, attachment, wherein like your bacteriophage, they will attach to a particular cell wall of a host cell. Again, we should consider the concept of specificity, wherein naghahanap ang ating virus ng specific receptors from found in the cell wall of your host cell. That's the very main reason why there are several viruses that can be found among animals that does not cause any disease. However, when humans are being exposed to this virus, we manifest this condition. Typical example, your COVID-19. Researchers show that coronavirus exists among animals. However, they do not cause diseases among these animals. Pero, pag ang humans na exposed sa coronavirus mo, particularly your lung tissues na may receptor site compatible to that coronavirus, we are now being infiltrated and infected by these viruses. That's why we are now developing manifestations of diseases, of this particular disease, this COVID-19 disease. That's the very main reason why, because of the concept of specificity. After attaching, we now proceed to penetration. Dito nagkakaiba. Unlike your bacteriophage that simply injects their hereditary characteristic or their DNA or RNA, ang penetration for animal viruses is like the concept of a Trojan horse. Pumasok yung Trojan horse na buo. Nandoon sa loob ang mga soldiers. Same is true with animal viruses. Buong papasok ang animal virus sa loob ng cell through the process of phagocytosis. Kakainin, i-engulf ang buong cell, ang buong nucleocapsid. Nucleocapsid because inside the capsid exists your genetic material. Now we proceed to the process of uncoating, wherein this capsid will now start to disintegrate, releasing this viral nucleic acid sa ating intracellular level. Now, this viral nucleic acid will take control over the host cell, dictating what will happen next, which will be basically your biosynthesis or the production of different viral pieces. Now, at this point, there are two options for your animal viruses: to proceed with your lytic cycle or to proceed with your lysogenic cycle. Let's first discuss your lysogenic cycle. Meron tayong tinatawag na latent viral infection. What am I trying to say with latent viral infection? Latent viral infection are usually limited by the defense system of the human body or our capacity to phagocytize and the number of interferons present or that are being produced by the infected cell. Sir, ano pong ibig sabihin ng latent viral infection? If there will be an opportunity wherein your immune system will be weakened, manifestation of the disease will happen. However, if your immune system is adequate enough, this particular virus will not cause any manifestation of a particular disease. they are dependent to two, our ability to phagocytize. Ibig sabihin, adequacy ng WBC natin. Once our WBC is adequate, hindi magkakaroon ng disease manifestation. Number two is this production of interferons. Just a brief review from our concept in anatomy and physiology. When a particular host cell will be infected by virus, sasabihin niya, there will be no chance. We will die in the end. But as martyrs, I would like to save our neighbor cells. Ganyan yung kanilang thinking. That's why we need to produce interferons. These interferons will cover adjacent cell, inhibiting the attachment of your new produced bacterial particle or viral particles from your lysed host cell. Both your interferons as well as your WBC count will be the determinant when this latent viral infection will occur. A classic example of this is your shingles. The causative agent of your shingles is the same causative agent with your chickenpox. Once na nagkaroon ka ng chickenpox, they will say that you are technically or you have technically developed an immunity for herpes zoster virus. However, if there will be a time that your immune system will be down or that you will be immunocompromised because particularly you are having chemotherapy or treating cancer or radiation therapy, or you might be of extreme age, ibig sabihin mababa ang immune system, shingles might occur. This is a type of a latent viral infection wherein the herpes zoster virus simply resides within the body and it will just wait for a time that you will be immunocompromised. Kaya siya tinawag na latent viral infection. On a latter stage pa lang siya nagmamanifest ng disease condition. The second option after biosynthesis, ayaw kong pumunta sa lysogenic. Punta tayo sa lytic cycle. It will proceed to the process of assembly wherein the produced viral pieces will be assembled and packed together having a particular covering or your capsid. Afterwards, it will cause the destruction of the host cell. Now, proceeding to the sixth process, which is your release, wherein the virions will now be released from the host cell through your lysis. The escape of these virions through lysis, the escape of virions will happen, some of them through lysis or other in the form of budding. Nabanggit ko na to kanina. Ano ba yung budding? Para lang siyang the viral cell that was now produced will be going on the extracellular environment wherein it will now be enveloped. Ano yung magiging envelope natin? Contents ng cytoplasm where they basically were packed or contents of the nuclear membrane where they were basically produced. Either dun sa dalawa na yon, it might serve as an envelope to your new produced virus. Next is your oncogenic virus. Pag sinabi nating oncogenic, oncology, it pertains to cancer because this particular viruses causes cancer. Examples of them are posted in your PowerPoint presentation. You have your Epstein-Barr virus that causes nasal carcinoma, Burkitt lymphoma, or B-cell lymphoma. You have your herpes virus. Alam natin yung herpes virus causes different viral infection. However, it might also cause Kaposi sarcoma, which is a type of cancer common among AIDS patients. Also, you have the hepatitis B and C virus that causes hepatitis B and C. However, it can also cause hepatocellular cancer, cancer of the liver tissues or liver cells. Next is your human papilloma virus. Especially our female audience, you know this. This virus is your causative agent for cervical cancer. You also have this retrovirus, a virus similar to that of your HIV, which causes a very rare type of leukemia. I'm talking about your T lymphotropic virus that causes your T-cell leukemia. Yamang nabanggit na rin natin, or now that we have mentioned your human immunodeficiency virus, let's talk about human immunodeficiency virus or your HIV. Its genus is lentivirus and its family is your retroviridae or your retrovirus. That's why, ang tawag sa mga gamot in order to combat AIDS or the causative agent of AIDS, which is your HIV, is your antiretroviral drug. Okay? Your human immunodeficiency virus are single-strand RNA viruses that causes AIDS. So don't be misled. The disease condition is AIDS. The causative agent is your HIV. These viruses binds to cell with CD4 receptors, which are commonly found among T cells as well as your macrophages. Sure, teka lang. T cells at macrophages. Yes, you are right. Based from your basic concept of anatomy and physiology, both of these cells are important for your immunity. T cells, okay, are basically cell-mediated immunity. Your macrophages as well facilitate phagocytosis of different bacteria or different foreign objects. Now that you know that they infect both T cells and macrophages, alam na natin na ang end result ng HIV ay pagbaba ng immune system. That's why, what's the problem among patients suffering from AIDS? Mababang immune system. They are easily being infected. That even at times, a simple case of pneumonia might lead to their death because they are immunocompromised. The other types of your viruses are the following. Mimivirus. Pag sinabi natin mimivirus, they basically mimic bacteria. Having that said, they are generally larger in size because they will contain a double-stranded DNA. But on top of that, if you can remember, a characteristic of a virus is that it will only contain either a DNA or an RNA. This is an exemption. Meron na siyang double-stranded DNA, but also it can have fragments of RNA. That's why it's larger by appearance. Researchers show that mimivirus causes pneumonia, some strains of pneumonia. Next is your plant virus. From the word itself, these are viruses that infects plants. Basically, this virus can be transmitted through vectors. Pwedeng madala ng insects, mites, or nematodes, or even by planting infected seeds or cuttings or even tubers. Or the tool that you are using to facilitate planting is infected of that particular plant virus. Next is your viroids. Don't be misled. Iba ang virions sa viroids. Ang virions, complete. Ang viroids, incomplete. Viroids does not have a capsid. And usually, these viroids infect plant cells, causing different diseases of plants by interfering with its metabolism. And the last is your prions. Ang prions naman, if your viroids are basically naked fragments of single-strand DNA, ang iyong prions ay fragments of protein lang na usually causes infections manifesting through neurological diseases among animals. Common dito yung tinatawag nating scrappy, wherein your pet animals will keep on banging their heads sa fences, sa walls, in order to relieve that severe pruritic experience. Yun yung cause, your prions. Okay, let's wrap it all up. To summarize our discussion, pinag-usapan natin ang acellular microorganism. We've basically defined acellular as something that lacks the basic structural foundation of life, which are your cells. We have also discussed why we need to include this in our study for microbiology, primarily because they are microscopic in nature, that causes significant physiologic changes in their host cell. We have provided 5 characteristics of your viruses or specific to viruses. Number 1 will be, it should only have either a DNA or an RNA, with the exception, of course, of your virus. They should be unable to reproduce or replicate by their own. They cannot divide through meiosis, mitosis, or even binary division. Number 4, they lack genes and enzymes to produce their own energy. That's why number 5, they only depend on their host cell. We've talked about the different processes wherein this particular viruses replicate. We've discussed your lytic as well as your lysogenic cycle, among, of course, with your bacteriophage. As defined, bacteriophage are viruses that infects bacteria. Also among animal viruses that infects animals. You have your different other viruses, namely your human immunodeficiency virus, which specifically targets cells with CD4 positive or CD4 plus receptors. You have the viruses that causes cancer, your oncogenic viruses, your virus, which is the exemption of our first provision. It contains DNA plus fragments of RNA, making them the largest. That is termed to be virus because they mimic bacteria. Your plant virus as well as your prions and viroids, which basically lacks some of the characteristic of a virion. With that, this ends our third series of our discussion, particularly tackling about acellular microorganism. I hope you all stay safe. God bless everyone, and let me remind you that learning is always a fun experience.